rust mites. The whole surface of the pear will look like a rust. The quality will go down. And this is with respect to strawberries. Tomato, we never had problems of mites on tomato. But in the last 15 years, right in the month of winter, the tomato rust spider mite starts. As it is, we don't know whether to eat tomato or not because of pesticides. This is the chili mite in polyhouse. They were originally Polyphagus tarsonimus was considered a greenhouse mite, and if you colored capsicum are being cultivated, green capsicum are being cultivated as a protected net, and mites are a severe problem. And one of the characteristic features you can see on the left side, top left, is the downward curling of the leaf and undue elongation of the petioles are excellent symptoms by which you identify the mites. This is on cucumbers, again protected. There are two crops which are coming major in protected cultivation. 60% is English cucumber and 35% is colored capsicum, and in English cucumbers, mites are a major problem. Now this is with respect to okra. I don't want to tell anything. Marigold. Marigold is 27% of our flowers in this country is only marigold. And Ganesh Chaturthi function, Sudha weds Rajanish or something, and the whole decoration you are doing, and it is full of mites. And not only that, they can cause severe allergy into this. This is on jasmine. Jasmine mites, you can see that this is called as a kind of a disease which is, people don't fail to understand. And this is important. Plantation crops you have seen in tea. One of the main reasons why our tea. This is the coconut mite. Highly microscopic. And when it came, people thought all kinds. Last 25 years we have tried everything on earth. Only thing we have succeeded is we have not been able to manage it. So today every coconut coming in every corner, whether it is Bangladesh or anywhere it is selling. Now the red spider mites are also going into many other leaves. I will not go. Now silk, as I said, is one of the most. We have got 5 different types of silk: mulberry silk, tassar silk, muga silk, and there is an oak tassar and eri silk. So I think yesterday the silk shawl that were given were tassar silk. But in case of mulberry silk, as I said, it is one of the most important because nearly our 70% of our silk is mulberry silk, and we cannot afford this kind of loss due to mulberry. Now, pesticide resistance, if you just look, you can see that these are some of the very new molecules, and already the resistance levels have reached thousands of folds to different set of chemicals on different crops. And there is going to be cross resistance. If you indiscriminately apply, other pests are going to this thing, and also the predatory mites will be destroyed. Now very briefly coming to this, excellent opportunities are there for management of the mites using the predatory mites. Coppert, one of the most wonderful organizations that is there in Europe. In Europe, most of the mites and other pests are managed, thrips and other things are managed in the glasshouse using the predatory mites, Amblicius, and there are many other species. This is the Amblicius. And the one on the right side is the one which comes on the grains. These are predatory mites which feed on the mites which come on the grains. I don't know if somebody has put this kind of a thing. I'll just go into this. These are all called the storage mites. I will leave the things with you guys. I'll not go into the details of what they are, but there are different kinds of predatory mites that are coming into the house dust mite. This is what I see. Assyria Chennabaswani. Chennabaswana is the father of Indian ecology. He is the one who started when nobody was there, and he is the one who identified the dust mite. It is named after him, called Assyria Chennabaswani. And you can be sure 60% of our allergies are basically coming due to this. And hardly anybody. One of the things we advise people is don't put carpets in the house because they are a source of dust mites. And if you are having problem of asthma and still you are using these kinds of things, you have a game that's going on in your house, and we don't advocate at all. And no matter in this, especially in urban centers, no matter what you do, dust is going to accumulate. Whether it is you touch your car after one day, there will be so much. You can be two things are there on it. One is Staphylococcus, another is the mites. I don't know how these fellows introduce this. They are, I'm not going to house dust. These are the dust mites. So sorry for that. I don't like these kinds of animations. But there are also parasitic mites. When I was a child, I don't know how many of you are remembering, we used to go to school and children used to come completely with shaven head and with sulfur all around their, I don't know whether Daksha has seen in his early part of life, but they used to come with in government schools, completely bald head with sulfur all over smeared onto this. Even when they go to Tirupati and many other places, you can see people put a paste of haldi on top of it. Many times, these are those days when unhygienic conditions, these kinds of mites become. Now this is one thing called the Varroa mite. We have two species, Varroa destructor in North India and Varroa jacobsoni in the South India. Varroa jacobsoni is not a serious problem, but Varroa destructor is a very serious problem. I don't know if it's there in Canada, but it's going to be all over in the temperate regions and in New Zealand. In USA, the loss runs into billions of dollars. One of the reasons for colony collapse is this. And that is caused by a mite called Varroa destructor. It is there in Kashmir, it is there in Punjab, it is there in Himachal. We are also hearing that it has been introduced by movement of beehives into the Tamil Nadu. So today what is happening is in beehives, wherever there is a pollination, we take the beehives. In the process, we may be transmitting many of the, this is the another group of mites which I will be telling, which are the parasitic mites. What happens is these are called as the follicular mites. They lodge themselves in the base of the hair follicle. We don't even know why these kinds of blisters are coming, and suddenly people have the habit of scratching, and then in the process these follicular mites go into different places. And you can see that these are the follicular mites, extremely microscopic and very easily contagious. And if you don't really wash your clothing, bedspread, somebody has sat in this thing, it is not washed in hot water, you can be sure that this mites. So the thing is, mites are not just a problem. See, there are only two few things. The more smaller the organism is, more powerful it is. So these mites, okay, you can see the kind of damage that they have caused onto the, this is with respect to the, on the dogs. These are all some of the animals which are getting affected on the streets. And what happens, every housewife and every owner, they don't want to maintain a dog, but they would like to give a food on the, their doorstep. So because they are the gods, but you don't take care of them, and incidentally, when they come and sleep on your corridor, shelf, doorstep, cage, it is getting dusted with mites. And you don't know what kind of allergy is going to get, and some of them carriers of what? This is a Demodex follicularum. This is the kind of damage. You can see the right side, at the base of the hair follicle, the number of mites that are accumulating. And these Demodex are one of the most severe causes for these kinds of, and 90% of the doctors are not able to diagnose them. They will simply give, okay, you apply this cream, you apply that cream, all kinds of makeup creams are coming. Everybody is going to, क्या बोलते हैं आजकल, हम सैलून पे जाते हैं, some men go to some, what is that called, makeup something, parlor, beauty parlor, I was the word actually. Beauty parlor में कौन आते हैं, क्या करते हैं, पता नहीं है, but you can be sure that is also one of the places where these kinds of infections spread, because even in salons. In fact, earlier you see the hygiene. When we went to salon in my house, you cannot enter the house, you have to take bath outside and leave the clothing outside and then come inside. That was the kind of hygiene that was practiced. Today, it is very cool to go and then hotel, restaurant and then take bath. And by then, how many places you spread all this, you do not even know. And considering the insecticide resistance, and most of the doctors are not aware of the ectoparasitic mites. So, this is the kind of damage that you see, the way the follicular, this thing is being picked. Of course, they're very simple. If you don't unnecessarily meddle with your skin and other things, 50%, most of them are amenable to sulfur, and simple sulfur and many other ointments can take care of this. I'll come to the, this is with respect to the, on dogs. I really don't know whether one should have pet dogs. I don't know. Oh, sorry, I think I was going again back into this. This is with respect to some of the lesions that are coming on the skin. So on the neck and many other regions. You may be reclining on the sofa cushion may be having these mites, and then in those places you get the allergy. Sometimes it comes on the skin, especially some of the mites only lodge between the fingertips and the toenails. So these are some of the mites. And just because they are microscopic, does you look at the architecture, they are like baton war tanks, absolutely loaded with all kinds of defense mechanisms, so much of sensory setae and many others. These are suboptic mites. They are a different group of mites. I wish some of our veterinary people were here to understand what kind of a thing that we are talking about. And another group of mites come only on the hairs. You will see some person always hair fall is there, they look sick, and then when you go to the follicle, you will have these kinds of damages. Now another small group that I am coming are the ticks. The ticks there are two groups of ticks, soft ticks and the hard ticks. The soft ticks come under Argacidae and the hard ticks come under, soft ticks come under Argacidae, hard ticks come under Ixodidae. And these mites, the female mites, they are so tiny and they will be there in the grass but somehow latch on to you, and once they suck the blood, they increase 25 to 50 times in number and they will be loaded with blood. It is not even easy, you cannot just squish them by using your toe or anything. They are so hard sometimes. And unless what we do in case of dogs and other things is, you will light up a matchstick and burn them or push them into the drain, and otherwise it's very difficult. You know how much to get rid of one dog tick it costs today? The same fipronil costs 2,000 rupees when it is administered to get rid of a dog tick. So many pet dogs are dying due to tick fever. So ticks are becoming one of the major... Talk, I stop. So this is the kind of engorgement that is taking place. Of course, this is the Argacid soft tick, and they are also responsible for carrying some of the most dreaded viral diseases, Texas cattle fever that is caused by Bufilus, and there are cycles where 1 year cycle, 2 year cycle, 3 year cycles. Western world has studied a lot. In India, unfortunately, there is no collaboration between. We are having a meeting on phytophagous mites. Nobody participates from the veterinary side. They have a participation, and most of the times we published a small paper. Of course, I was the one who initiated this work on HLMR. In the month of Bakrid, hundreds of sheep are slaughtered. And many of the sheep will be having so many of the ticks. And when you slaughter an animal, the ticks will fall onto the ground and immediately latch on to the person, the butcher. And these are transmitting what is called as Crimean Congo hemorrhagic virus. It was found in Lahore, it was found in Rajasthan, it was found in Punjab, and many of these came from the border area. Especially a problem in Muslim countries when mass-scale slaughtering goes on because of Bakrid and many other festivals. In the Western world, slaughtering and packaging and marketing of meat is very hygienic. हमारे यहाँ तो रोड में butchering होता है, रोड में उसको लगाते हैं. And in that butchering, there will be also dogs. How many cattle and how many... I think we are one of the most resistant species on earth. Otherwise, we should have been wiped out by now. Considering the kind of animals and other things that are there in this. Now, what are the opportunities that is with respect to the study of acarology? They are microscopic, molecular systematics. There is a huge work to be done. If a zoologist asks me what I should work on, I will say immediately work on ticks and mites, because there are no experts available. There is a tremendous gap between the knowledge and what the societal need of this particular aspect. There is a perceptible shift going towards biological control, especially protected cultivation. Mass multiplication of many of the phytosid mites are a problem. Organic and natural farming is requiring for the management of mites with ordinary sulfur you can manage, most of them, things. And there are wonderful botanicals which are available. Synthesis and evaluation, don't confuse insecticides with acaricides. Acaricide physiology is totally different from insect physiology. So there are opportunities. Greenhouse gas effect, if somebody wants to know what are the pests that are going to be severe with global warming, mite is a classical example. Overseas knowledge, harness and importation of potential natural enemies. At least somewhere in the West, some people, even in the West today, whether it is Smithsonian Institute or many other institutes, there are no experts. Many of my friends are going and do the taxonomic work because they are also finding it difficult to hire persons. Commercialization for biological control, economic analysis of mite damage. Today we are having a lot of problems with date palm and oil palm, date palm and arecanut. Very difficult to work and there are no experts. The threat is rapid development of very high pesticide resistance. I would put it way above everything. We are just unable to control mites. For example, in case we thought black thrips of chilies, everybody said black thrip of chilies, but how much of mite was there? Nobody knows. Lateral flow of high pesticide resistance from polyhouse to the open. Elimination of predators due to indiscriminate. Not only the loss in climate change is due to mite. Mites will survive. Predatory mites will collapse. Unscientific and indiscriminate import of plant material without quarantine. We are importing dime a dozen. This country absolutely has no rules for importation. You tell me tomorrow what is to be imported, which plant, 24 hours, you don't require Amazon. It will be delivered on your doorstep without quarantine. And I asked somebody why most of the invasives come very close to research institutes, because the first person to do this kind of mistakes are the scientists. Entry of alien mite species, I am completing in 1 or 2 minutes. Global warming likely to favor phytophagous mites and while reducing the biocontrol. Future line of work should include the future of the project is dependent. Somebody should, at least I would request to the AZRA in the recommendations to go that I talked to the DG when I submitted the QRT report as well to the 75% of the country is not covered under the ICRP mites. We are simply celebrating double the farmer's income, triple the farmer's income. Let us avoid this kind of bogus terminologies. Real commitment is required in having the project go into more states. If 2/3 of the country is not covered, then what is it that we are going to do really with regard to this kind of problems? Quarantine remains the weakest link, and in the globalized world, transboundary diseases are going to become a major problem. So also transboundary mites and other things. When you import a plant material, and if it's a torsionomid and an acirid mite, how are you going, a quarantine man, without even a proper microscope is going to tell these mites are there? IP of mites under protected cultivation depends greatly on IRM. There is no understanding of acaricide resistance either in the world or here. Whatever happens with cholinesterase or glutathione transferase or the neuromuscular junctions of new molecule is way different in mites as compared to insects. Their hemolymph is different. Their physiology is different. It is like comparing the fishes to birds and saying that both are same. Commercialization opportunities are there. Botanicals and lot of work to be done on pathogens which impact mites and pesticide residue, related pesticide residue. When we are talking about mites, pesticide residue people won't come, vegetable acrylic don't come, fruit people don't come, floriculture people don't come. There is no meeting of the mites. Today I wanted some horticulturists who are working on floriculture to be there. Vegetable people who are on protected cultivation should have been there. Animal scientists should have been there. Like we have become so much in silos that we don't think through this. Thank you very much. I think I have finished it on time. If there are any questions, I would be too happy to take.
Dr. Mr. Kumar has delivered a wonderful lecture, and he has, made us all think about these, small creatures, which we just ignore from day in and day out, because they are right there, even in our sofas where we are sitting properly, he is scaring us there. But anyway, I invite any questions or, any kind of information you want additional, we are, welcome to ask our
Always have excellent vacuum cleaners to suck out the dust from your sofas. Wash your pillow covers and bedspreads and other things in hot water. You don't have to wash. Dip them in hot water and dry them. That's more than enough. Next, sir.
Dr. why ICR or Indian government is not providing research funding or hiring people, bacteriologists in the university? Why it has not happened?
Sir, the research funding in this country has come drastically low. And in case of ICR, if I tell, we had 5 KVKs, 10 KVKs, 15 KVKs, Krishi Vignan Kendras earlier, which was supported by ICR. In the last 20 years, it has increased to 730 KVKs. And the entire KVK money is coming from ICR research fund, number one. So 33% of our research fund is going to Krishi Vignan Kendras. Then we have coordinated research centers which take about 30% of the mango, citrus, rice, wheat, sorghum, etc. And the establishment cost is taking place of under 25%, and the real research grant that is available is hardly there is a scientist has sufficient funds to travel 100 kilometers, no support for travel, no support for equipment. This is the hard truth. Many of the agricultural universities do not have money even to pay the salaries, but they want a political that they want to divide the universities into. In each state has got 5 universities, 5 vice chancellors, 5 registrars and no money to pay even salary to the staff. It is an absolutely fiasco and policy paralysis. Yes.
But in fact, in Odisha of late we are seeing the rice barnacle mite in a very big way.
Yeah.
So it has become a very common problem, particularly in Kharif season towards the fag end, where dry climate is seen or the dry spell is observed. In fact, honestly speaking, sir, we are not in a position to give the real control measures to the farmers, which they will be having a tangible effect. By the time they are seeing these symptoms, everything is gone. So, maybe about a couple of years back, we had been to a particular area, 30 acres of land completely spoiled by this, this pinky species. So, can we have some viable management practices, sir, which you can recommend to the farmers besides these miticides, so that they can get rid of this type of problems?
First is sulfur still remains as an option, one of the best acids. My second recommendation is please pass on the message to the, even I can tell the Odisha OUAT Vice Chancellor and the Department of Entomology to have a small acarology unit going, because rice is a big crop of the Odisha and you cannot afford to let your guards down. Third one I would like to tell is, in the month of February 1st and 2nd, in collaboration with ICRISAT, possibly Dr. Joshi also have invited IRRI, and cement and cotton, we are having 4 crops breeding for multiple pest and disease resistance. Mite resistance has not been really done. We have neither evaluated the germplasm. Probably mites, the panicle architecture many a times can hold the key for what type of the mites and many other things. But the problem is you cannot breed a resistant line for mites, one for the loose mud, one for the blast, one for the blight. We have to have multiple disease resistance. So the whole theme of the discussion will be multiple disease resistance. But I would say at this particular point of time, even a use of water, sulfur and many other things should be more than enough to manage many of these mites. But I am not an expert on rice, so I will not comment. Somebody must have come from rice. I don't know if they are there. I wish Joshi was here, because he would have been the best person to answer the problems. 2 things in rice. One is nematode, another one is because dryland rice is directly proportional to nematode. Dryland rice, the mites is going to become a problem. So I would probably, the best person to give answer is, please note this question. and push it to Dr. Joshi. I hope that he will be in a better position to answer this.
Thank you, sir. We have been using these sulfates with the sulfur compound,
Not of
but as
control.
but it's a therapeutic action. But can we go for its prophylactic application? Can we recommend?
Among the acaricides, the best thing that we are recommending is, of course, I should not be telling oomite. That is from that comes from Dhanuka pesticide. Oomite is one of the best acaricides.
Oomite, sir, we are taking care.
Yeah,
we are
oomite,
recommending.
among the acaricide, all the AICRP centers recommended was, among the acaricides, oomite was the only acaricide which was doing, of course, fenpromoxin or some other new
yes, sir. The new
have come.
molecules.
But unfortunately, they are so expensive. Second one, mites will develop resistance in no time.
Yes.
So you have to be very careful about how do you use this. I would request somebody to take up seriously this kind of postgraduate research work instead of simply telling trichoderma, we are also multiplying this kind. It is also required. It's also the requirement of the professor to give that kind of like yesterday Dr. Joshi was telling. His professor through the whole thing. 3 months he worked in the library and came out with the proposal, and even after 30 years he is still into rice. So we need that kind of postgraduate research.
So last question, sir. We observe this pinky has got relation with the sea throat disease. This rice panicle mite and sea throat, it has got a direct correlation. So wherever we are observing this one, the sea throat is also coming up in a big way. Can you please
because there are many things which are favoring. Leafhoppers, mites, thrips, they all progressively increase with one set of weather parameters. So the other thing, for example, generally when we had discussion in ICR, every year everybody used to discuss drought in June. I used to laugh and Dr. Ayyappan used to ask, why are you laughing, Krishna Kumar? Then I told, sir, in horticulture crops, the problem is excess rain and not so much of drought. And in field crops, the problem is more of drought than excess rain. Of course, excess rain this year in rice also and many other crops is a problem. In future, you are going to have long dry spells provided by and followed by very intense precipitation. So these kinds of things we have to calibrate, understand, and it is changing from year to year. So there are many things that need to be looked into. Sir, you wanted to ask something? Any students here? Any students? I would like students to interact. आप इतना पीछे क्यों बैठे हैं? हाँ. No, you hold it like this, horizontal to your.
Yeah, sir. So I have 3 questions of myself, Dr. Rajshekhar. Sir, yesterday when we met first time after long time, you said you have conducted a meeting on chili crop pest. Chili is being an export crop, and in India, in all the chili growing areas, chili is much more damaged by black thrips, also
mites and white flies.
So among these two, the result, we are not knowing what is coming first and what is coming later. That the thrips are coming first, or mites are coming first, or when mites are there, thrips are dominating, or thrips are dominating the mites. That is not known. And one more thing, the farmers when visited the Kandukuru, Gunturu, all the chili growing areas.
Hold it like this.
When I visited the farmers in the southern Andhra Pradesh and all chili growing areas, farmers are not knowing what to spray, because when mites are there, insecticides are sprayed. When insects are there, miticides are sprayed. When both going alternatively, so whether that is causing a resistance to mites or thrips. So one more question.
What was your first question?
First question is, which is dominating?
Okay, I'll come one because I forget the sequence of question. First one is, when the temperature is dry, hot, with less humidity, thrips will dominate. when the temperature is hot and humidity also builds up, mites will dominate. In fact, mite is considered as a greenhouse pest, typical classical high humidity, high temperature, mite will dominate. Dry temperature, red spider mites and thrips will dominate. But in case of Polyphagotorsonemids, high humidity, high temperature, Polyphagotorsonemids will dominate. Andhra Pradesh, we had a review, 60 sprays were given to chili, 60, and sprayed every 3 days by our NAM group.
Yeah.
And 90% of it was unwanted. Of course, we told them. So the question of it is, if the petioles are getting elongated, think that mite is becoming dominant. If the upward curling is becoming more, it is. And black thrips you can see because it's going to dance on the flowers.
Yes.
So the question of it is, what we suggested is, between one insecticide, go for one botanical essential. Even today, neem soap, pangamia soap and many others are excellent answers for sustainable management and preventing the resistance. Instead of simply going, there is no silver bullet for black thrips. There is no silver bullet for mites. Only thing we can manage is, even pangamia oil is one of the wonderful source for as a synergist. So we only recommend one insecticide, one botanical, one insect.
So the second question is, I was working on pheromones and semiochemicals for a long time. For mites, any pheromones can be there or any attractants can be there.
Pheromone, even if it is there, it should be used only as a monitoring device. And where billions and billions and billions will multiply. You know, Polyphagotorsonemids can multiply to billions in a matter of 4 days. So if you say I will trap it with pheromones, it is impossible.
Thank you, sir.
And a student, yes madam. You come front and speak, so no problem. Give the mic. I would like the mic also to go quickly. Sir.
Sir, actually digitization is growing faster way. So whether any database is designed to identify the mites?
Yeah, there is quite an amount of good database. We have also done for at least 30 to 40 mites under the acarology for a molecular database using MTCO1. But what we have done is only tip of the iceberg. What we have done is tip of the iceberg. There is a whole world that is waiting to be discovered. That's why I am telling the molecular biologists and acarologists and others should join. Young SRF should come forward to say that, okay, I'll do. For example, Jacobsoni and Varroa. Varroa Jacobsoni is not at all problematic on honeybee. Varroa destructor is a deadly mite. Both of them look alike. So molecular biology can, especially for invasive mites, your digitization will help tremendously. So the first slide that I showed you itself was on the MTCO1 to say that we can do. Yes, but we have done something, we have miles to go.
And second is any sensors or sensor type device is designed to identify these mites?
Not yet. Not
okay,
yet.
it is all.
Not yet. Madam.
Sir, how can farmers monitor early signs of resistance?
How can all of you have to learn one thing. The way you hold this is horizontal to the mouth. Most of them hold like this. Your voice will not be heard. Hold it horizontal to the mouth. Yeah.
Sir, how can farmers monitor early signs of resistance in mite population before it becomes severe?
One is, when you spray and it doesn't get controlled, farmers immediately know, kuch nahi ho raha hai. Second thing is, there are excellent methodologies by which you can tell the resistance the workers can do. Unfortunately, what is happening is, lot of cross resistance is also taking place. Now I will share with you some of the information. I launched the product Coragen about 15 years back. When we introduced Coragen, 100% damage control of the brinjal fruit and shoot borer was there. Today, even after you spray Coragen, 50% of it is not getting controlled. So, in the case of red spider mites, we have resistance across any molecule, only the level of people are working on that. That's why I said this requires human investment, scientific investments. You cannot just generate data without scientific investment. Okay?
Sir, one more question.
Yeah.
Are there any successful models from other countries for sustainable mite management that could be adapted here?
See, the good question. most of the data that's coming is from the Western countries. Western countries are cushioned. They don't have this kind of a temperature. Tropical regions, the problems are 10 times, 15 times more. You take capsicum, you take chili. Capsicum is 5 times more susceptible to mites and thrips. Chili is 5 times less susceptible as compared to capsicum. They don't have the kind of selection pressure as much as what we have. So as a result, their modeling and whatever that happens cannot be directly applied to the tropics. So having said that, there are a few models that are available and that has been done in US Bangalore. I would request the institute like this, some of you should participate in the acarology meeting. Wonderful people are coming from across the globe and across the India to share the knowledge. All ACRP centers are coming. So discuss with it and learn whatever this thing. But I would be a little more interested in all of you also paying more attention to parasitic mites, dust mites, household mites, biocontrol mites which are can be useful for biocontrol, and also we should be really focusing now through the veterinary on ticks. They are deadly. They are deadly. There is a disease called monkey disease in Karnataka, transmitted by a tick called Haemaphysalis spiniger. What happens is, first they infect the monkeys fall dead, and the farmers take their cattle to the forest, and this Haemaphysalis spiniger comes to latches to the human being, and in a matter of time, to 5 days, 10 days, they die of fever, and internally they are called as hemorrhagic fevers. It's called monkey disease. Yar Shumaga, somebody had come from Soraba and those belts, they will know. So I was also talking about there is another mite called as the rust mite, which is transmitting a different kind of a disease in tomato, which we are able, that's called russet mite. Very recently reported as a vector of one of the diseases. Assyria Kajani, Dr. Lava in ICRISAT first time, he is the one. First time in the world showed Assyria can be a vector of the pigeon pea sterility mosaic. So that basic work today after, he is now the head of the division of IITA in Africa and voted as the best scientist of IITA 2 years back. So if you make good scientific contribution, the world will recognize. Am I taking more time or please?
Thank you, sir.
I think maybe the last question.
Last question.
Sir, if time is there, myself is S.N. Mahapatra from nematology. We know that there are some mites in soil. That is from, I don't remember their generic names,
Okay.
but they are from suborder Mesostigmata or Astigmata.
Okay.
They are having a predatory habit on nematodes. They feed on nematodes. Okay, so they remain in soil. So, sir, please educate me whether all the mites they remain in soil or they are remaining everywhere in air but what you told.
Every organism has got its own niche and they will be there in their niche. So the mites which are there on the hair, mites that are there on follicle, mites cannot live in soil. So like in your case of your nematodes, where 99% of the nematodes are not parasitic and many of them are saprophytic nematodes. So also a number of nematodes are there saprophytic and they are external indicators of soil health. So all mites are not parasitic.
Yes, sir. But there are some mites which feed on nematodes.
They can be there, but they don't, there is an association, but they cannot be considered, we have seen, they cannot be considered as parasitic in many insects.
Okay, sir. Sir, then the mites infecting the crops, whether they are remaining in soil or where do they remain actually?
They are multiplying on the plant surface. For example, rust mite will be there on the fruit, the leaf mite will be there on the leaf. So there are where they have their niche and that's where they survive. For example, chili mites will be there only on the undersurface of the leaves.
Okay, sir.
Okay.
Okay, sir. Sir, last one. Sir, in household, if you spray the sofa and the carpet with surface sanitizer, will that kill mites?
The sanitizer and cleaning only is on the exposed surface. The size of the mite is 1 micron or less. They can go to the crevices and they will be there in millions.
Okay.
समझ गए ना आप?
Yes, yes.
तो ऐसा है कि you cannot eliminate them. More the hygiene, the better it is. Best is to wash a little bit with alcohol or fumigant, these kinds of things. These are going to be the problems, especially in the urban areas, sophisticated. A person who is working outside, for example, mechanic, cleaner, in the motorbike shop and all, they never suffer from asthma or cold because they are genetically they become resistant. But those who are there in air-conditioned rooms always have allergies. So this is also a question of how our body adopts to this.
Right.
Okay?
Thank you.
Thank you so much. Thank you so much.
Okay, thank you very much, Dr. Krishna Kumar, for your fantastic talk on these mites, which has brought a lot of, discussion and, everybody wanted to have more information on each one of these items here, because they are present universally everywhere. There is no place where they don't hide. So we have to find ways and means, and also, as you said, we have to extend our research more deeper into this area so that we get more answers for all these questions. So everybody is raising there. Thank you very much.
Thank you, sir. Thank you so much. It's always a pleasure when you interact after your presentation. Without taking much time, I would request Dr. Vidyasagar to just share his expertise on the smart and sustainable IPM for red palm weevil, regional innovations and global strategies for palm. The red palm weevil on a date palm is a Indian introduction. They call it as an Indian insect. If you go to Saudi Arabia, it is called as Indian insect, and everybody is scared. And this red palm even went to many countries through ornamental palms. We did not bother about ornamental palms. It went into this. Severe loss is being caused on date palms, and the entire Middle East is worried about it. There are seminars on date palms only for the purpose of management of red palm weevil. He was a consultant in FAO, he was in Saudi Arabia, he has vast knowledge associated. Even in our own country, red palm weevil is making a comeback, also on coconut, and also is also becoming a problem on many other palms. And though there are pheromones, to a great extent it has not been. And we are very eager to listen from Dr. Vidyasagar on how to manage red palm weevil in the current scenario. In fact, when I went to Saudi Arabia, the Saudi Arabian agriculture minister had a separate red carpet for us because he always wanted to discuss this. So that's the importance the given Middle East gives into this. So the next one also you keep it open so that next time the person comes immediately the slide should come on the screen. Okay? Please.
Good morning. I am Vidyasagar, and before I go to the talk, I would like to extend my thanks to Dr. Anand Prakash, my very close friend, and I wish that Dr. Rao recovers very fast there. And yesterday night, most of us met him there, and I again extend my best wishes for her speedy recovery. Now, this is a topic I have chosen to share my experiences with this almost like 30 years of my experience working on this red palm weevil in the Middle East. And now it is a global pest there. In 2016, we have organized an international symposium where 18 countries have participated who are suffering from this red palm weevil, whether it is on coconut or date palm, but mainly it is date palm. In MENA countries, that is, Middle East and North Africa, it is a devastating pest there, and the farmers are losing heavily on that one there. So I will quickly go through this thing there. See, the overall about 9.4 million tons of dates are produced there worldwide. The top country is Egypt, followed by Saudi Arabia, and then UAE, like that, Algeria, then Iran, Iraq. But you don't see India in this map. So whenever I was, in a meeting there, chairing or something like that, so I used to ask them, why you are not including India here? Because we have small countries from Africa, but not India. They said, you don't have processing plants. They say, unless you have a processing plant, we cannot include it. In India, what we produce, you can see this figure there, 171,000 tons are produced. But in the FAO data, they have even 70,000 tons of countries producing, but what they do is, they have a systematic way of, processing the fruits which are harvested. But in India, what we'll do is, my previous speaker has told, we don't care much of hygiene. So similarly, see what is, processing is not a big thing there. What they do is, after collection from the field, you have to put it through some water bodies, clean it there, dry it, then pack it, and then label it. So, these are the things which we have to do that, and we have tremendous potential in India. And India, importing $264 million worth of dates are, imported in India because it's a very important fruit, nutritious fruit, and it is also health fruit. So, we have to use this one there. See this one there. Here you see the 490,000 tons of this one, tons, this what you call dates fruits are imported, dried date fruits are. In India, what we produce is only the fresh fruit. We don't produce the, completely. There are 4 stages in the date fruit. The last stage is the tamar. So that is the one, which is dry fruit. But we don't produce because of the climate conditions there. But fresh fruit, yes, we can certainly produce. Global spread of this red palm beetle. So, globally, it has spread from India, what they say, because more than 125 years, it has been reported in India, and you can see here in this picture there, so India in red color, from there it has gone to the Middle East, and then it spread in the Middle East. From there it has gone to North Africa, from North Africa to the European countries there. All surrounding the Mediterranean, they have this red palm weevil. But of course, in MENA countries it is the date palm, but in Europe it is the Canary Island palm, which is a very ornamental and very important for them. Biology, ecology, and symptoms of damage. Of course, this red palm weevil, which is having 2 life cycles in a year in Middle East under the conditions there. Actually, what I have found during my research there for the last so many years is that though the temperature outside varies so much, the plant inside maintains certain kind of constant temperature inside. It's the vascular tissues and all those things. But still, there also during winter times there is variation in the temperatures there. So because of the climate change, because the theme of our symposium is climate change and its impact on various things there. So what we found is why it is increasing there, because there is a change in the climate there also, where the cold temperature days are much less than what were about 30-40 years back. What they used to say is more than 50 years back there, the cold temperature days are much more, the number of days there. So they have done some research there and they found, yes, there is variation in that one there. So the climate is, even during the winter times, it is becoming more and more warmer. So the damage is devastating. First, it is a cryptic pest, it is hidden inside the stem there, and once it is inside there, you cannot find it there until the damage is very extensive, and you can see that a lot of oozing, then there will be, finally the crowns will fall. What is the socio-economic impact of this pest. See, as you know, food is the moral right of all who are born into this world. That is, Norman Borlaug. So we cannot deny anyone the food what they need to do. Similarly, of course, the last one many people will not agree because Norman Borlaug was completely against using organics only. No, because what he said, you can also have as a component for the overall benefit of the fertilization of the plant, but not only depend on organic and say no chemical fertilizer there, because as we see that we have seen in our Sikkim state, they made it as organic for 10 years. Later we found that the yields have been drastically reduced, and now they are trying to see how to improve those things. The loss of the money for a farmer, we have estimated, trying to see that even if the percentage of infestation is 1%, because infestation, what we go by is one palm is removed out of 100 palms, that means 1% we can say. So like that we calculated, you can see the figure, 59,000 million tons only in Saudi Arabia every year they are losing if we don't control the pest properly. Current management practices are listed here. You can see here prophylactic methods, chemical methods, mechanical, chemical, stem injections, pheromone mass trapping, biological methods, microwave techniques, precision agriculture. And now what is there is we have to see how artificial intelligence can be introduced into these components so that we can use them very rationally without wasting anything there. See, most difficult part of this pest is detection, because since it is a hidden pest there, it is inside there, and the earliest stages cannot be identified from outside. Even a trained eye finds it very difficult there, because no symptom is seen there, because in a microscopic hole it will go inside there and it will go on, tunneling inside there. So, and then multiple larvae will be there inside. These are the methods, like thermal cameras, sniffer dogs, visual inspection. These are the three things which are available here. But you see, when I was there in one organic farm, which is nearly about 25,000 hectares, we introduced the sniffer dogs. And we were successful actually. For about 3 years, there was no infestation at all in that garden. Acoustic methods, these are there for a long time. It has been, improving, more and more sensitive sensors are available there. It is improving there. Still, we have not reached a stage where we can definitely say 100%, the infestation is detectable. So IR cameras we tried there, but here also it was so encouraging there. For about 6 months we tried with these things there. But it's also only partially about maybe 50% we can detect, but remaining 50% will go unnoticed. Some, Italian companies have developed a X-ray technology where what they said is they were huge kind of, sensors and all those things, X-rays, and you have to pass but this can be used mainly for quarantine purposes. If somebody is wanting to export some 500 palm trees, because the adult palm trees can be exported anywhere there. They are such a wonderful tree, Yeah, even a 20-year-old tree can be transplanted anywhere in the world. So for such things, this will be useful there. But again, very expensive proposition. Sniffer dogs in the field, how they are using it there, training and other things are there. Actually, Spain has really, has done a wonderful job there. Because in Canary Island also, Canary Island palms also, they have this issue there, serious problem there. And that is also their heritage, they don't want to lose the Canary Island palms. So they are trying to develop this one there, sniffer tax, in their nurseries, because they have the nurseries, huge nurseries are there, from there they sell the trees, and each tree may be costing about 2,000 euros or something like that, very expensive. Of course, this is a kind of a case study there in one farm, how we can do it there. I have demonstrated here. stem injection, see, it is there for more than 50 years there in India. From there we have started it, and that is the same thing we are they are doing also. The MENA countries also, they are using the same stem injection, but in addition they are trying to use some fumigation methods there which are not very successful, but some people still insist it can be used in some conditions there. These are the injection methods there. They have recently, about 10 years back, they have a high pressure in stem injection. They say within 3 to 4 hours stem can be injected with some 5 liters of pesticide there, and they are trying to use it there. It's a little expensive because since the governments are funding that one there, many farmers who are having large farms, they will buy this kind of equipment. This is the one, electronic method. They tried there in Italy there, and one company was insisting this is a very useful method, but it's a very huge equipment. One has to move in a small farm, is very difficult there. Only government can buy that one and use it there. But again, selectivity is reduced there. You don't have that wide application of this particular technology. What they say is, they will identify and also kill the insect inside there because of the temperature. It will raise the temperature to 42 degrees. So what we ask them is, can it be really useful when it goes to 45 degrees sometimes? Then probably the tree itself may be affected, the yield may be reduced there. They have no answer because the next year yield will be the same thing or not. They are not very sure it there. So that's why this was not a popular one. Of course, this is the one, which is used most of the time in most of these countries which are suffering from red palm weevil infestations. This is the pheromone technology there. So there are 2 components which are added, and now they have gone into the nanotechnology with the same pheromone technology, where they have reduced the amount of pheromone with the same efficacy there without, this one. This the area-wide pheromone technology there, where you will have that pheromone traps located at different places in a designated manner there. And this is a male aggregation pheromone which attracts both males and females. That's the big advantage for that. In many other pheromone technologies, we have the disruption and all those things. But here, this is a male aggregation pheromone, and where both sexes are, in fact, females are more. If you, for about 3 years, we conducted a study and found that more females, about 60% females and 40% males are attracted to these traps there. Only thing is the bait. The bait is a must there. Only pheromone alone will not attract. Pheromones have to be added to that. So by adding pheromones, what we are doing is a kind of, a renewable product. Means, every week we have to change it there. And in high desert conditions where wind is very high, within 3 days also the liquid bait dries up. So they have to replace it, which is a very tedious process there. Physically it has to be done. But in recent time there is electron trap also which they have tried, electric trap. So where no food is required, no bait is required, they say it is equally good. Of course, see, the main reason for so much of infestation in Saudi Arabia was large areas which were having date palms have been abandoned because of the prosperity, economic prosperity due to the oil wealth there. So in the eastern province where it has started, most of these people who are having the ancestral properties of 10 acres, 5 acres, they just left it there without cultivation. So no irrigation, no cultivation, nothing. When they have abandoned those gardens, these insect pests have resided there. They were like reservoirs, they were acting there. Nobody cares about it there. Nothing was there. The trees were falling down. Nobody attends to that one there. From there, they were spreading everywhere, See, they have demonstrated in recent time more than 3 kilometers to 7 kilometers it can fly and, spread. So each, insect, adult insect can lay more than 300 eggs there. And it will lay not one day, it will lay over a period of 7 days. So the spread is very easy there. So we have proposed a program where they have removed nearly 500,000 palms in a matter of about 2.5 years there. It was a huge project there, and they have bulldozers, we have dumping yards, we have spraying methods so that it doesn't, whatever is escaping from there will not spread to the healthy gardens like that. So in that, different kinds of, methods have been used. Of course, this I will skip there. Now, RPW risk analysis, So there are 3 stages. The offshoots have to be taken care of, because offshoots are the method from there it starts there. If mother palm is having an infestation, even from the nursery where they are collecting, may also have some adults sticking to that one there. When they go to a new garden there, it may affect that garden also. So they have to take care of the offshoots. Then after transplanting in the new garden, also they have to take care of them. Maybe some soil insecticides can be applied there. They can be given a spray for both pathogens as well as the insect. And finally, regularly they have to see the prevention, and also for monitoring they can have the traps. And also after having, if any trap is receiving a red palm weevil adult there, they can start praying and other things, looking for that one there. So these kinds of things will help them to have clean gardens. So quarantine is another issue there. You can see this one huge trees, nearly province to another province, 500 kilometers, it is transplanted in the open like this. So if there are any infestations in these palms, it's like a kind of, very serious matter there. We told the people there, and now quarantine process is done by the agriculture department. They have the agriculture officers who are supposed to give a quarantine certification after proper inspection, spraying, prophylactic spraying, everything they do. Then they will have also a label. A metallic label is attached to each palm that it has been inspected with the date and all those things. But many people don't follow it. These are the methods we have to, worldwide, we have to implement. See, countries affected by the pest should take suitable measures to ensure pest-free planting material. Because whenever we have organized an international conference in 2016 in Rome, and there we have told the people there, all the country people who are representing, like their director generals or the deputy ministers who have attended, that we have to have a comprehensive system where good methods are used for collection of the planting material and also the treatment of the planting material there. And we have also published a book. Some 20 experts who are there have written different chapters there, so that is made available to every country. And also we wanted to have an international a body there in Egypt for all the MENA countries like Middle East and North Africa, who will be like a nodal agency for a global referral system for this pest there. And things were happening there, but not fully. It has been made functional now. This is about the plant quarantine methods. I will skip this one there. No, I am going backwards. Sorry. Yeah, this one. This is a very important one there. Here, you can see that one there. In 1992-93, the spread of this was about 6.7%. And in 5 years' time, with integrated pest management, we have been able to bring it down to 1%. So in recent times, it is less than 1%, is okay. If it is more than 1%, one has to act on that. Challenges and the current gaps. Early detection, of course, yeah, it's very important. Then sniffer dogs use pheromone trapping, pheromones with dry traps is also very important there. And biotechnology in RPW control also we have to explore. That is going on actually. dsRNA methods and RPW antennal olfactory system studies and gene silencing is also going on. Actually, the child I created in King Saul University is working on that system. How to we can do the gene silencing so that they can be controlled in a economically and also ecologically better way. Not use of so many chemicals. So nanotechnology for Bavaria bassiana. Bavaria bassiana, this fungus was found to be good biological agent. Unfortunately, in those desert conditions where the temperatures go very high there, how to spread it there is a major problem there. And if we can have some nanotechnology where that pathogen can be protected, then I think it will give us good results for a longer and sustainable period. IPM models should include methodologies for different ecosystems. One ecosystem will not work in. For example, the ecosystem in Kerala for coconuts, where also dead palm beetle is a major pest, will not work for desert conditions because there the architecture of that tree is different there. Its harvesting times are different, and like that and its climatic conditions are different. So we have to evolve methods which are suitable for that ecozone. Of course, we try to do some remote sensing there. So we have a chair in precision agriculture. Dr. Patil from Dharwad was there with me, and we tried to collaborate and see how best we can do that. Can we make use of this remote sensing and say, okay, this patch is having more of palm weevils are there. For example, more than 1% of palm weevils trees are having palm weevil based on the color of the canopy there. So we tried there, but still we did not get the results what we expected there. But a lot of research is required into that. Yeah. Biological control methods, many people know that one. They're going on for the last more than 50 years or 60 years. Lot of things, and especially nematodes, they tried very well there. We also tried in Saudi Arabia, but that was not successful there. In a laboratory, yes, in a laboratory you can get excellent 100% result there with these two species, Heterorhabditis and the Stringer nema. But I don't think in the field it was successful. At least I tried in about some 20-acre area plots in 3 places. All the 3 places, in the first 1 month we could see some result, but later on it just vanished. These biological control agents, how the adults will become, when they are affected by these fungi. And of course, see, we have to use, the modern technologies in the precision agriculture. We need to use the drones there for inspection of the tree. Back I use of this one. And of course, this is trap. This is without any food. No pheromone is required here. The food is loaded there. The pheromone is loaded, but there is no food or anything. Wet is not there. It's a dry one, and one need not go every week or something like that. One can keep it there, forget for 1 month, and go after 1 month and collect the insects and again clean it up and then allow it to sustain. But this was from Italy, and in Europe they are using it there, but in the Middle East and North Africa it's not really so popular because one is very expensive. I think it's about $450 for each trap, so that way it has not picked up much. Natural products, repellents, attract and kill methods. These are 3 areas where more work is to be made. We tried to do some with the plant products. We got some good results in the laboratory there. We had some publications, but we need to have more data on that. This genomic and bioinformatic approaches for RPW is also needed there. So we have this, not only the Rhynchophorus, about the insect we do lot of DNA studies and all those things, but we also have to do some work on the endosymbionts and pathogens. this also we need to do there. So once we get enough data based on all these things, then we can devise methods, how we can make the approach and target the pest. Of course, this is about, endosymbiont bacteria and how we can do it. In fact, King Abdullah University of Jeddah, there is a university which is having a lot of faculty from Cambridge University, and we had a collaboration with one of the guys there and one Arnab, and his PG student was doing, actually postdoc student was doing, and he found there are some symbionts are there, and we wanted to elaborate on that one there, and subsequently some people are doing there, but I have left by the time. Smart and sustainable and region specific one is required there. Solutions perceived are, risk has to be first assessed, and we have to eliminate those things there. And here, this is the most important one there, a matrix of activities for sustainable IPM, where research is done by the universities, institutions, private companies, and then they have to share with the development agencies, public as well as private organizations, and extension. Unless we have a strong extension wing, whatever research is done will not reach the farmer, who is the user, So that is the thing there. And farmer participation, we can have, yearly or biannual, we can have farmers exhibitions where, progressive farmers are invited and the knowledge is shared with them there. And we can also hear their problems there, specific problems to that region, so that we can solve it, or we will try to find solution for that one there. That way, so four things, they have to interact properly, then only we will get some good results. Conclusions. Adequate support should be provided for developing, testing and adoption of new innovative methods of IPM management. Align scientific advances with on-ground applicability with artificial intelligence. Now this is very easy, Nowadays people are preparing papers also with artificial intelligence. Probably they may not give much, but still people are trying to do that one. But we can certainly use them for making use in the integrated way, where different systems are used there. So we can have the expertise, from this one, the whatever is available, nook and corner of the world, what is available, we can retrieve through AI means and then incorporate it into our systems there. We need not take totally from whatever that AI gives us. And then, of course, national and international quarantine lines have to be improved. Entomologists gathered there. And then I was one of the invitees. And they said, you may be knowing, Dr. David, who has worked on nematologists, but a lot of work on palms also he has done. He is from Florida. So they said, Rhynchophorus ferruginous has been discovered in California, Long Beach Island. Everybody was, really, I should tell you, they were all, in huddle, and they said, what is going to happen there, are we having that one there? Because California is, pride of America, the best medzul, the largest date fruits, the top number one, is from California. Okay, that is not native to that, but from Morocco. What has happened was, from Morocco, they have taken 14 palms trees, the nursery, in 2014, planted in California, Conchilla Valley, and they found excellent results, and that is the top producer of that variety, medzul. Okay, so that's scary, If the red palm beetle is coming to California, means the entire thing will be devastated. But luckily it was palmera. Wrongly identified, who have 121 belt for 1 year. Hoffman, chief entomologist, because I organized in the USA one California, because I want to what actually happened there. They don't share. Little bit, farmers will be scared the answer. Okay.
It's not a question, but just my clarification.
No, yeah, welcome. See, here I am sharing, my experiences. So, yeah.
So in fact, we are also having a palm areas. He belongs to the palm area. So there is a large acreages of palms. So we are suffering from this space, then rhinoceros petals, and also there is a darker report. Moving around our no tree about a distance.
Pheromone varies from company to company because some people give you 90%, some people give you 70, 80%, because so something like that. The micromembranes, they will have release and high release. This help to two kinds of pheromones. So, Keptica is the one which we have started. Dr. Cam Olschlager, the founder of this pheromone, when he was in Simon Fraser University, his student, Dr. Rebecca has discovered that, this particular pheromone worldwide. That was in 91. And they started trials in UAE, initial trials. Then Saudi Arabia invited. At that time, I was also there. By that time, they have not developed the trap best. We have so many designs, We tested about 20 traps of this there. Finally, the bucket trap is the cheapest one there. Now, this pheromone, believe me, for 5 years I was working there. And after 2:30 when I returned from the field and my house, so many times I found my coat with weevils around, two weevils, going around, because I was handling the pheromone, distributing that one there. As few traces are on my this thing. And once I wanted to put, I have a small farmland, I want to put some traps. I called Olschlager, I sent a mail and he sent me 10 pieces of these pheromones. They were in my house, I could never put in the field. For 6 years or 7 years, I was getting weevils into my kitchen. They were kept there in a closed one plastic shield, but because even the small traces, if they are going, the plume will go in the floor and they will come there. So the traces, these small traces. So now, one Indian company has started selling this nanotechnology. One Subharan was there, who was my colleague, junior colleague there. So I gave him idea. See, if in India we have a lot of people who can do the nanotechnology, because if you go to Europe, it will be very expensive. So we have to shell out a lot of money there. I said, in India we can do it quickly. So he has worked on that one with the Indian Institute of Sciences and another group together, three people. They found it that the nanotechnology is possible there. See, 600 ml, 600 milli, this one there, less than 1 gram. 600 milli is there in a sachet. So we can reduce to 300 with the same result. See, farmers in the beginning, in 1999, when a company in Kerala was selling this pheromone in India, they were selling at 1250 rupees one sachet. One sachet. Too expensive. How many farmers per acre, per 1 acre, some 5 acres, 5, and this has to be changed every 3 months because it will dry, You have to check it against the light and see whether it is a pheromone or there or not physically. So means 4 sachets, 4 into 1,250. How much? 5,000 rupees for 1 acre. This is too much, It's not that worth there. So we have to go for the nanotechnology, smaller one there. So only 100 ml, 100 milli, not ml, less than 0.1 ml, 1 gram will be sufficient for 1 year actually if it is properly released. See, that slow release also in tropical countries is very good. For other countries where the temperatures are lower, for example, in North Africa, I think probably during winter season the temperatures will come down lower and lower in the night time especially. So there it will stay for much longer time. In summer it will go in 15 days also.
Any other questions? Quick questions.
Yeah.
Sir, have you applied a light trap method? Sir, usually insects are attracted to light. In your presentation, I could not see the light trap was applied anywhere.
Light trap?
Yes, yes.
Yeah.
So
light I have not put because it will be too much. No, light traps.
No, we can have in intervals like 500 meters if you are putting constantly light trap for 1 week. So possibly the population could be reduced for some extent.
Yeah, I will tell you. See, this light traps are put there in Saudi Arabia, many places. Not for this, for the other one, that Orictis. Because that also is a problem there in young plantations there. For that they put it there. And also, a lot of these farms are having cattle. So cow dung and other is available there. So they could breed there. Some of these species there may not be our species. Other species are there, and the 2 species are there, which are there. Plenty of them are there. We do get this red weevil will not come to the light trap.
Okay.
Yeah, you put it on nothing, not a single one. It's not attracted.
Any other questions? Just give me 1 minute. In the case of
Sorry, because I am feeling very cold. That's
No
why I am
problem.
not able to really speak.
in the case of India, where in coconut and many other places this is a problem, we advocate a couple of things. That is, when you climb the coconut, don't use the sickle marks to continuously put the sickle marks on the stem where the egg laying takes place. You can easily spot the plants which are about to collapse, and that is the time when dispersal of the grubs takes place. So immediately, as soon as the trees collapse, make sure that the area around that place, you collect the grubs and destroy the plants so that they don't become breeding grounds. Also give a lot of care for movement of ornamental palms which house a little bit of many of this. With regard to the pheromone technology, though many persons are talking, I have a feeling of 2 things. One is, pheromones many a times may become counterproductive by bringing the infestation from very far off areas into areas which are not so much into the place, and unless you really destroy all that they are getting attracted, you may introduce. So there are many issues that have to be looked into. Entomopathogenic nematodes, we are looking just to sprinkle around the infected plants so that all the larvae that fall into the vicinity can be destroyed. We will do one thing. I will request, due to paucity of time, after he finishes his talk, those who are interested can please one-to-one have a discussion.
Yeah, sure.
And thank you very much, Vidya.
Thank you very much. Thank you for giving this opportunity.
Thank you,
the organizers, Dr. Panda, thank you very much.
Thank you, sir. Now I call upon Dr. A.K. Dubey ji, agar hoga to 20 minutes, for 10 minutes for discussion. Because I don't want to curtail the discussion. And just 10 minutes, and with a little more focus on Venetia and of course others also.
Good morning to you all, and thank you for providing me the opportunity to present a paper here. I have been working on whiteflies for last 25 years, and today I am going to summarize some of the things. I wanted to present actually the whiteflies of and I have come to know that many are confused with invasive species, and some of us want to know how to identify them, how they look like, and how to differentiate them. So there are many slides, around 70 slides. I will try to finish within 20 minutes. Thank you. Just go ahead. So what are the whiteflies? Whiteflies are the common sap-sucking insects. They are less than 0.1 mm in size, and they are called fly, but they are not the true flies. Like dipterans we refer as a fly, so whiteflies are not true flies, although their name is given as whiteflies because of their white appearance in the adult stages, and their body is covered with the dusted mealy wax. They are very similar to aphids, scale insects, and cilids. And the important thing is whiteflies are not identified based on the adult structures. For identification of whiteflies, we use pupal cases. So this is the only a group of organisms where we use pupal case instead of adult for the identification. The major reason is whiteflies are more reliable characters are in the pupal stage rather than adults. Adults are monomorphic. So one to subfamily level we can identify them, but genus and species it is rarely. And they are injurious to plants by directly sucking the sap and indirectly by transmitting the plant viruses, especially the family Geminiviridae. They secrete honeydew in all the stages except the egg, which increases the sooty mold on the leaf surfaces. This is the life stages where we find the adult will lay the egg, and egg it's hatching in nearly 1 week. We call it crawler because it can move on the plant surface. After that it will lose the legs and antennae and become atrophied and sessile. Within one place the second and third instar will develop, and they are not movable, only the first instar will move, and they are occurring on the undersurface of the leaves. So within the place first instar settles, up to fourth instar they will be on that stage only. This is fourth instar, see dorsal view is a lateral view, and the adult emerges from the pupal cases. So this depends on the different stages, and different species have different biological cycles. Some complete within 1 month, some goes up to 2 years in the European countries. These are the SEM structure of adult morphology mouth part rostrum, through which they feed on the leaves. These are the chemosensory sensilla. These are the detectors because some of the whiteflies are the host specific, and they will be feeding only a particular host plant, and these are the labial sensilla, and this is male genital organ which is useful in the identification of the species in some of the genera. These are some of the habitus images. This is a spiraling whitefly, Alerodicus dispersus. This is another Venet Alerodis megapapli. This is a new species which is not yet described from Uttarakhand. This is greenhouse whitefly, Trialerodis vaporiorum. This is again the same species. You can see this one and this one in mature form in early stages. This is Emilia sonchifolia and the venter. So, based on the secretion of this wax pattern, we can say what are these species, and this could be an identification character. When the agricultural students are confused, we just tell them to send the photographs, and based on the secretion of this wax pattern, we can tell them what could be the possible species. This still here you can say this species described new. This is Disampancta, pest in West Bengal and part of Odisha. This is a new species from Great Nicobar Island, not yet described. This is also a new Schwartzian genus. This is a Cinnamomum and see the Aleomar. See, all of them are having different type of wax secretion pattern and very identification character. In whiteflies, we are having 4 subfamilies. Out of the 4 subfamilies, the Alerodini and Alerodicini, these 2 are the mostly abundant, and this one is Bernaini is a fossil, and Udamoxilin is represented by only 7 species. Some of the whiteflies are common in India, in that we have greenhouse whitefly, Trialerodis vaporarianum, sugarcane Neomuscalia bergei, and Alerodobus barodensis. Similarly, in jasmine, cardamom, betelgeuse, citrus, this is the most common is Vaglumi, citri, and Alerocanthus spiniferus. Spiniferus is best in the European countries. Then cotton, we are having problem of Bemisia tabasi and Babul Acadeliodis rachiflora. This species is abundant in the western part of India, rachiflora, especially Recently, there are 9 now 10 invasive whiteflies are reported in India. Within a span of 8 to 10 years, we are having invasion of nearly 10 species. So all of them are reported as invasive, but mostly 5 to 6 are only invasive, because some of these they are just reported as invasive, are not causing any economic loss in India, and we don't have biocontrol agents or any other control measures for them. Here, what could be the reasons that has led the invasion of these species in India? The one may be the climate change. Second, the trade practice, which is free between the countries. When suppose like guava and all we are importing or exporting, usually with guava we are having 2 or 3 associated leaves. Those leaves are having pupal cases, and then they get spread or introduced to new areas, and due to high climatic favorable conditions such as high humidity and temperature, they stabilize in the coastal areas first and then spread to the middle in the northern part of the countries. Then export-import operations of our planting supplies. Potential natural enemies are not available in our country. Then second is the insects population is very highly increasing whenever they are introduced to new and favorable climatic conditions. Here are the whiteflies. These 4 species belonging to subfamily Aerodicini, and this around 6 species from the Aerodicini. So majority of the pest species are belonging to this subfamily Aerodicini, which is mainly distributed in the North and South America. That means northern part of North and South America, New World, and these are the old worlds. In the old world means except the North and South America, in which we are having only the major problem because of the Bemisia tabaci. And recently the Aleotachelus annoni is also reported as an invasive, but it is not causing any problem on annona or guava trees. We are having main problem of tabaci. Trichoides and Procosus is now damaging the guava trees, this one, Procosus, and Trichoides on chili and other species. Here are the list of species. When you look into that, Alerodicus dispersus, it is from the first affected Menihates esculenta. This is mostly fascinal coconut species, coconut trees. Alerodicus rugia operculata. This is infesting mainly coconut. And if you see coconut, most of these species are confined to monocot host only. And Alerothecus trichoides on the capsicum annum, that is chili, and acacia and lucena. This is a fodder crop. But we are having problem in coconut, and multiple species are infesting together in the South India, Tamil Nadu, Kerala, Andhra Pradesh, and part of Karnataka. Now recently a survey has been conducted in Kerala by myself and a team, and we found in Kozhikode area, farmers have dropped cultivating coconut tree, and they are shifting to the fodder crops because of the loss by these whiteflies. They are not active flyers. Okay, I have to go to next one. Here. What could be the most climatic condition will be the fluctuating throughout the year. These are the whitefly population. Once build up, it will fluctuate. When there is heavy rain, the population goes down. Within after 1 or 2 months, it will come again. Then high atmospheric humidity, that is favorable condition for the whiteflies. When humidity is more, it is around 60 to 80%, the population goes high. Similarly, the temperature and rainfall will decline the population. The host plant health, if the plants are healthy, the whiteflies will be propagating more. They have enough source for the eating and multiplying within the host plant. And they are found application in the insecticides, they are mostly not effective because most of the whiteflies, they are feeding only on the underside of the leaves. So when we spray the insecticide, it is not reachable to them. Second, most of the whiteflies, they are having heavy, thick layer of waxy coating. So whenever we apply insecticide and it reaches, it will protect, the wax will protect the insecticide contact with the animals, either in pupal stage or in the adult stage. So these insecticides are not much useful. And only thing we can use is the better way is the natural enemies to control them. Here we have done some analysis of whiteflies. When we look at the geographical extension of this and distribution of genera, we find the maximum number of genera are described from the oriental region. That means the whitefly is maximum having the diversity in oriental region followed by the neotropical region. Similarly, the species are also more in the oriental region. here, the species, when you see the oriental region, they are having the maximum number of diversity compared to Australian, Australo-Oriental region, Pacific Islands, Miami, then Mediterranean region, Western Palearctic, Neotropical and Nearctic region. The maximum number of whiteflies in the oriental and second is the Nearctic. But again, when we looked into the distribution of the species, we find the one subfamily, Allirodini, only occurring in oriental region and this Allirodicini. So here are 10 species of Allirodicini subfamily are reported from this old world. They are all invasive from the neotropical regions. So that is the region we are having the most invasive species in India. They are all came from the new worlds, that means North America. Here we have taken a project, that is 2017, not 27. So 2017 to 2024, we had a project on whiteflies of Andaman and Nicobar, which I wanted to discuss here. So here, Andaman Nicobar we have taken because it is very isolated island in the Bay of Bengal, and it is around 1,400 kilometers from mainland India and 200 kilometers away from Indonesia and related islands. So we wanted to know the genus species so far not discovered in those islands, and finally we have adopted a standard methodology like collection of whiteflies we have collected from the islands where it is not reachable. We have taken boat, ships and reached to some inhabited islands, brought to laboratory and flights were processed. Finally, we could identify some of these species I will be talking about. This is the Andaman and Nicobar Island in the Bay of Bengal. So, this is the Andaman group of island. This is the Nicobar group island. This is the Andaman group of island. North Andaman is divided into three major islands like North, Middle and South Island, or this is a Little Andaman. Here is a 10-degree channel and this one is the Nicobar Island. So it takes around 24 hours to 48 hours to reach from this Port Blair to here, and we have only ship journey. Now the tourists and others are not allowed, only for research purpose we can go to the Nicobar Island. So we have taken a project permission from forest departments, visited to this Great Nicobar. And this is smaller islands here, this one North is Long Island and some Little Andaman these areas. So there are 572 islands. We could collect whiteflies around 30 islands. And then we have come to with some data. 1200 whitefly puparia were mounted. 25 species under 18 genera were recorded from these islands. 81 species in 40 genera were identified. And this found 56 species were new record to Andaman and 22 genera were new records. Altogether, 30 species in 8 genera are recognized as endemic to the Andaman Island because they were not at all matching in morphology with any of the species described from mainland India or the adjacent countries. So we made them this. Then there are 3 species like Euphori and Elleroclava bifurcata. They were also reported as new to India. 3 invasive species we reported here. When you see the population, this is a coconut tree, fully occupied with this Ellerodicus rugia perculatus. This species was described from America by Martin in 2004, and this is the pattern. When you see the coconut leaves entirely infested with this wax secretion pattern and shooting mold growth. Then this is the guava tree. I don't know this much infestation you have seen or not, but in Andaman we are having this is the pattern of this one, Hellerothrixus crocusus. I could rear this species for more than 2 months in the multiple locations wherever I get. Unfortunately, no natural enemy was collected. Recently, NBII Bangalore has got one, Garcia cubensis, and they say it is successful, but the percentage is only 5% in controlling this. So, unfortunately, this is also not successful. And Rugia perculatus, we are having some one Amitus species and one more Encarsia, but that is also not controlling this whitefly. And since the tree is very tall, and we cannot apply any insecticide methods here. Then one wild host is here in the creeper. It's called glory worm. Here also we are finding another species, Trachoides. It's a very severe, you see. And this species is using this host as an alternate host. And whenever there is a chili season, this will transmit from this host to the chili. This species is Neolitsi, known from Australia, and this was from Thailand. We also got it from the Andaman Islands after nearly 80 years. This is the mostly damaging species in India. Cotton whiteflies, we call it, Bemisia tabasi. And this is having a highly invasiveness because wherever it goes, it is having the highest number of host plants. It is a polyphagous. It can breed more than 500 host plants and damage implications are very high. It is having the capacity to transmit plant viruses causing diseases, and insecticide resistance is there. Life cycle is very short. Within 1 month it will complete one generation, yearly up to 12 to 14 generations. It has ability to adapt various climatic conditions, and it has got various biotypes. Biotypes concept was earlier like they are having several haplotypes and genetic variations are there, but morphologically they are similar species. But recent studies have indicated that some of the synonymized species, they are not the synonyms and holotypes, but haplotypes. They are not biotypes, but they are independent species. We are trying to sort out some of them, and 4 species are going to be retained as a valid. Now, the species identification itself is another problem, because most of the whiteflies are showing host-related variations. It depends on the leaf surface nature. If the leaf is hairy, whiteflies will have more elongate seeding, and the leaf surface are having a smooth leaf. The pupal surface will not have any indication of the seeding. Take for this is the same species, Bemisia tabasi. This is on tomato, where you find like brinjal and tomato when you compare are smooth leaf. When the leaf surface is hairy, we are having like this mimicry of the CT as a trichomes. And when the leaf surface is smooth, these cities disappear. They are there, but very small. We cannot detect. Only you see variation 1. And similarly, they are having variation in the occurrence of the CT in the asymmetrical pattern. Suppose one side, here if you see the earth abdominal CT is present, but other side it is absent in this case. So these are the another hurdles when we try to identify species with very limited populations. So because phenotypic plasticities are associated, we need to take them and to account that whether it is same species or different, because Bemisia itself is having more than 50 species worldwide, and most of them are identified based on the presence or absence of some of none of these species. This is a spiraling whitefly. I thought I should keep some slides for students so that they can identify easily. This is called spiraling whitefly, aerotic dispersal. In India, this was the first major pest in coconut, nearly 90s. And it is laying the egg in the spiral sequence on the leaf surface. You see, this is a wax is threads like a spiral sequence. They will lay the egg like a spiral. In middle, there will be some 60 to 70 egg laid by the female. This is the female, and this is a known name derived as spiraling whitefly. Adults emerges in 2 to 3 days, and eggs will hatch in around 1 week. Total duration will be for nipple stage 13 to 14 days. One generation will be completed in approximately 1 month. And this has got some natural enemies also, so it is not much, but in Karnataka, Bangalore, and some of the urban area, this is feeding mostly on the Bohenia trees, teak, Bohenia, and these are the major host plants for this species. And this is a pupal case, very easy to identify. 4 large pupa will be there and 1 in this region for total 5 pairs. And this is the drawing made by John Martin. Now, recent invasion of this species is Rugi operculatus. This species was described from Belize, and now first it came to Tamil Nadu in Polachi, then coastal states Andaman, Assam, West Bengal. Major plants are banana, coconut, oil palm, cashew, mango, ramphal, water apple, and jackfruit. These are the severely affected by this species in the southern India. The most easy to identify this species is the lingula. Genital organ is pointed and reaching to the posterior area, and this is on the coconut leaves, where you can see the clumps of wax. Fluffy wax will be there, with some of them are elongated, and this marginal area is having small bands of the tubercles. It's quite easy to identify. Now, this species is a Tetraeleurodis acacia. India, it was reported as invasive, but as I said, it is not invasive. It is just a new record for India, because it is not affecting any of the economic plant or commercial plants or having the value. Yes, I will come to you. So what happened? This species was collected on this sword bean in Maharashtra area. This is so many different sword bean and ramphal something. Here we are having intermingling of another species. When you look at this, there are 2 species. This is another species Eleurodicini, this is another species, and this is third species. So there is a mixture of 3 species interbreeding on the single leaf. This species was first described and then it in citrus from Brazil in 71. Thereafter it was not placed in those countries. Slowly it is moved to different countries like from Brazil to Puerto Rico, Florida, then California, Hawaii, and recently it last was reported from the Taiwan and then now in India. This is called nesting whitefly because the adults, they are very lethargic in nature and except bands are found on the wings, so it can be identified easily as a Paraeolirodis bendari. It will make a nest and the adults will be just settling down and feeding within the nest. And these are the eggs like grafts, and this is the wax nest. Inside pupal cases or immature stages will be there. Eggs are provided with a pedicel, very creamy like color in pale creamy color, easy to identify. The most important character is this one, the nest pattern formation of the in the pupal stages, and the wax nest is formed by the secretion of wax from the immature stages. Adults, very easy, the antenna will be thick compared to other whiteflies, long, and which is lethargic in nature. They will not fly until disturbed. So it's the eggs band, if you see, thick antenna, and this is wax nest, then you can be sure it is a Paraeolirodis bondari. The another species very similar to this is called a Minei. Both look alike, but the nesting pattern is like this, scattered little. It's not like star making, and this is not having the X bands on the wings. So both are the same, both are invasive, and that is the only way to identify. This is not having the except the pattern of band on the wing, and the one is having band. In pupal stage, they look alike. There is no difference in the pupal stages. Both species are same. So what we have to do? We have to just allow them to emerge the adult, collect the adult, and then mount the adults in the slide. This is the differentiation, bondari versus Minei. There is no band. And adult stage, when you see, Minei is having two long spur on the male adiagus, and bondari is not having. Only in the case of males we can identify, females cannot be used for the identification. This trichoides was first reported by myself in India, by myself and Dr. Sundaraj in 2015 from the Duranta. This is the ornamental plant in Bangalore. But we never expected after having this much infestation it will damage the chili. But now it is feeding on the chili within, after 2015 to 20 there was no report from chili. 20 onwards it is feeding on the chili. Sir, I will finish, just to give me 2 minutes. This is the pupal cap and very easy to identify, having this band on the submargin, this area, and here one pair of CT. This you see similar species, but there is nothing on the abdominal region. This is the crocosus. In India, it is feeding on Gava, but is in the Cuba. It was described in Cuba, first reported in Kerala in India. Now it is feeding maximum on the Gava plant, but in the European country it is feeding on the citrus. So we don't know why the varied preference is there in European versus India. Europe it will feed only on the citrus plants, but India it is not feeding on the citrus plant, but feeding exclusively on the Gava. And the flocuses, if you see, this is the pattern. It's the creamy puparia plus glossy wax. And these are the two species. This is flocuses. And this is trichoides, which I have shown earlier in the having the blend on the submargin. This is not having the bend. Both belong to same genus, and it's very easy to identify. So this is another species, Phyllory. It's found on the sapota tree. Then greenhouse whitefly, sugarcane. Yeah. Here, some of the whitefly species are intermingled on the host plant. In that, one is the Peleus minei and another is Nagarcoilensis. Nagarcoil is a place in the Tamil Nadu from where the species was described. But now in the Orissa, West Bengal and other parts of the central North India, this species is becoming pest on the Annona. Now they have reported that Annoni, Alerotrachelus annoni is feeding the pest as invasive, but more than annoni, I found Peleus nagarcoilensis is the pest in India. And the Ficus religiosa is infested with Shingella simplex. This species is native to India, but it is not pest in India. This species was described by Singh in 1931, but India it is not causing any damage. But recently it has reached up to Taiwan and European countries where it is severe pest of the ficus tree. And another one species we have got from Arunachal Pradesh. This is new record to India. Population is very heavy, but still I will not report it as invasive because it is not on any of the economic plant. It is feeding on the wild host plant, air climber. In Andaman, I will come back after invasive species. This is a new genus we described. Another new genus we described from Andaman. Third genus, Siraphilium, Covid Alerodis, now Dhritya Alerodis. Some species were reported as new. So all of this indicate these are the new species we are describing from Andaman. None of them are published so far. So this is another set of new species. We are writing the manuscript. Only this is published in this. So this was the species I described from the tea leaves, Camellia sinensis. And after that, the tea board has argument that why are you describing it will degrade our tea productions or tea import export or something. Then I said I got this species, so I have reported it. The next part is of your management. So I will facilitate you how to identify this species and detect which are the host plants are infected. You eliminate them. These are the pore-like structures, very easy to identify the people cases. Deluensis. What is the conclusion now? Prior to this species, 25 species in 18 genera were known from Andaman. We described 81 species discovered from the 40 genera, 56 species were new records, 16 were published as a new species, 5 new genera we described. Still 21 species are pending in collection for publications. I am writing, these are the endemic species list we made. Some new species, new genera. So what we have concluded from this species? The island, White Andaman and Nicobar White Plains, Andaman and Nicobar Islands, whiteflies are more closely related to Malayan region than to Indian mainland. When we look into the morphology of the whiteflies occurring in Andaman Island, they are not at all resembling to the mainland India. They are more resembling to the Malaysian region. So there is some kind of evolutionary origin and some kind of relationship when they have divided in earlier age and developed independently. More new and unique species may be present in the islands. As I mentioned, 578 islands are there. I could only survey around 30 islands. But other islands, there will be many more unique species and exploration is needed. Exploration could resolve generic complexities. How the genera like Bemisia, currently Bemisia tabasi only we know, Bemisia giffardii there, Pongami is there, so many species. But Bemisia itself, genus is not a monophyletic. It is having many complex of another genera. And Bemisia tabasi itself is not a one species. And genus Bemisia in India itself, it should be split into at least 4 to 5 new genera. They are not belonging only to same lineages. Early detection of invasive species and movement can help because Andaman Islands, whenever how the whiteflies comes, when they come, they first establish island, then coastal area, then they propagate, then migrate to the northern part of the country. So when we can continue survey in Andaman Islands, we can get early detection of the invasive whiteflies movement and their establishments. Economic loss we can minimize not by the synthetic pesticides because we use directly vegetables and fruits and spice crops. So insecticides are not recommended. The best thing is use the natural enemies. We can have the propagation units for Guadalupian cassia and cubensis, etc. So we can apply them and release in the fields. Eco-friendly methods are spraying plain water. 30 to 40% population can be minimized, and after interval of 2 weeks or 3 weeks, we can have second spray. Neem oil is the best. I have also tested it, and 1% neem oil is sufficient for 2 spray after intervals of 2 to 3 weeks. There is nimididine, nimidazole, neem-arc, neem cakes, many neem. Chloropyrifos also I used, T-pal also used. Michaelia Champeka and Persia macarantha dye was obtained from the leaf extract and bark extract was found useful. So those are the some eco-friendly control measures can be adopted rather than the insecticides. Management is very crucial until we detect the early stage of their establishment in the agriculture crops. Management becomes difficult in the later stage. So best way to detect them early, just based on the wax secretion patterns, which I mentioned before in the first slides. And immediately you just remove the leaves or adopt the simple water spray. Domestic quarantine methods can be strengthened. Stakeholders can be, or farmers should be just sensitized. When they are looking some wax secretion pattern on the underside of leaves, they can be told this could be a potential pest species, so eradicate them. Then avoid use of insecticide, use biocontrol measures. So these are the things. Thank you.
In the present scenario, anybody working on whiteflies is worth his weight in gold, considering the importance of whiteflies in the national scenario and the national economy. You are a friend of mine. You are there in forest research and some time or not. Positions which are one is we need a not there in Andaman, so that advise our groups to say, look, this is there in Andaman, don't bring it to mainland. This is there in mainland, don't take it to Andaman. So that will be one small recommendation.
Already published a paper in Andaman.
No, as a recommendation.
It was there in Andaman earlier, not here.
As a recommendation from the your presentation of this. Number two, gone are the days of even biotypes. We are talking of genetic groups in whitefields. First it was zero group, then wheat groups. I would be really happy if a real marriage of these two things can take place, because see, we can't go in search of a taxonomist to identify.
So we are remain like a is once the pupa becomes empty, it is just a head of people try to extract the pupa.
Take the limbs and do the this thing, MTCO1 based on the live.
Yeah, mitochondrial cytogram vaccine. I have shown one more slide where we are finding the intermingling of the other species. Okay, we are having to more than two to
very strong.
yes.
What is as a follow-up of this, immediately we need to develop for all these species.
Yeah, that is
very
required.
Rapidly.
That is required. But we need adults. We need adults.
Yeah. The parts. So when we say possible clustering of North Andaman, let it be done. There are a couple of thoughts. I am a great fan of Encarsia, Gudalopi, Haiti. experiences and many others. Is you can find white fly and Bemisia tabacco, demon transmitting DNA viruses. Their mutations, the entire subcontinent.
Thank you.
We'll move to the next. We'll not any questions can please come in the suggestion box critical to your work. Fibrosa. This area fibrosa.
Yes, sir.
And shall we break and come back? 3 minutes. Please assemble. It is very sorry. Please go back. Please go inside. अच्छा नहीं लगता सबको बोल देना अपने आप 5 मिनट में आ जाइए और शुरुआत करें. Thank you. Nice one. Who's the next person who has to make a presentation? You are requesting somebody who was not feeling well. Namaste, कैसे हो भैया? कैसे हो भैया? आप सबके अच्छे फिल्टर्स में थे, देखा 2 मिनट के लिए. अरे फोटो तो आपके साथ बताइए ना. I know, I met it. Yesterday, जाओ, you will never get that kind of a knowledge. All I say, 10 times more than this.
Sir, आपका
देखिए मेरा क्या है, कृष्ण कुमार
ବ୍ୟାଙ୍କଟା ଇସଲେ ସେ ବ୍ୟାଙ୍କଟା ରିପିଟ୍ ମାରିଲେ ଚାଲିଥିବ ରିପିଟ୍ ଚାଲିଥିବ ଆରମ୍ଭ କରି
मैडम यूटर मैम पर्दाटा
ସୁଆର ମୋଟାଟା ପକେଇଥାଉ ମୋଟାଟା ଯେଉଁ ଆମର ସୁଆର ମୋଟାଟା ସୁଆର ମୋଟାଟା ସୁଆର ଲୋକଟା ଇଏ ହୁଏ ମୋଟାଟା
are the students? All the students, please come front. Who are all the students? Please come front. And the questions have to be asked by the students. ऐसा नहीं है कि यहाँ लोग बड़े लोग जो 4-5 बैठे हैं वही सब पूछते हैं आप सामने आइए welcome back, and then we are making a small little bit of a shuffling where we request Sonali. Madam, Sonali Bendre is very easily to pronounce because Sonali there, but हम सोनाली कहते हैं बेंद्रे आ जाता है पता नहीं क्या. Anyhow, you are also as beautiful as her. Formulations of entomopathogenic nematode, heterorhabditis, the survival and infectivity, a wonderful field. Raipur पे हैं हम. You are in Raipur?
Yes, Raipur, Chhattisgarh, sir.
Chhattisgarh. You are a principal calling authority. I am coming to Raipur next month for the biotic stress meeting.
Welcome, sir.
मिलते हैं. So EPN and their survival and infectivity, an excellent field of where a lot more needs to be done. I don't know how many of you know, one of the first genetic sequences of the entire end-to-end 3' to 5' genetic sequences was done in some of these nematodes. They literally are like Drosophila for many of the aspects. And madam, please go ahead and
Okay.
you will be given about 7 to 8 minutes time.
Okay, sir.
And please allow 2 to 3 minutes. भैया सवाल सर ये बेल है आपके पास? ले आएंगे तो बेल. So it becomes easy for us to monitor. And madam, please go ahead.
Okay. Okay. Thank you, sir. Myself, Dr. Sonali Devne. I want to present one work of a very small work.
मैडम आपका माइक नहीं एडजस्ट हुआ है तो ऐसा लगता है हाँ हम नहीं सुन पा रहे. ठीक है.
This is my students, PhD students' work, and and here we tried to make formulation of EPN. That is very, very important because we know that EPN have great potential to kill insects, and it is very important biocontrol agent for soil insects particularly, and we can control foliar insects as well. So EPN have majorly 2 genus. One is Heterorhabditis and second one is Stenonema, which with the bacteria Xenorhabdus and Photorhabdus in their intestine. And although EPN have proven to be successful in as biological control agent against many insects, and their efficacy is also very good, but in large scale it is impaired. The use is very impaired. Why? Because of the short life cycle, which is due to their need to maintain biological activity, their high oxygen demand, and particularly it is very sensitive to high temperature. That is the main stored material for successful commercialization. Minimize the mortality is important. Second one, the infectivity, correct infectivity, very important. And one most important thing in nematode, that indigenous strain is very, very important, because it is already accepted in that soil fauna and environmental conditions. So here we
the slides are not there. थोड़ा देख लो भाई. ठीक है, आप you call switch, you will do.
Okay, and so for that we all know that greater wax moth is the best host for the rearing of and mass multiplication of EPN. So we have collected this Galerium melonella from the honeycomb, old honeycomb particularly, and through the artificial diet we mass reared this Galerium melonella, and and collected soil samples from the 3 different zones of Chhattisgarh, that is Northern Hills, Bastar Plateau, and we have the EP techniques after after the one sent it to the NBIR Bangalore, and we found that all the 6 strains are Heterodendrogenes indica, and the virulent strain is was isolated from the mango Raipur. Through white tract, we have isolated all the EPN strains. This is the healthy larvae in left hand side, and right hand side this is the EPN infected through the Heterodendrogenes indica. Then after harvesting, this is the inoculation technique. We continuously filtered that that the culture nuclear tissue culture flask. So formulation, the first sponge pieces which was washed through thoroughly in the top water and autoclaved population from the sponge degrees centigrade, the 6 different stages after inoculation is 45 days, 60 days, 75 days and 95 days. And we observed that at the first 2 intervals, means 15 days and after 30 days, 10% and 87, which grade with the increase is the 'PSC' or pathogen mortality, that is 80% at 24 hours duration. Larval mortality at the same duration was 72% at 60 days, and at last 2 storage, (75 and %) respectively. So in the next formulation, it is hydrogel. Hydrogel we prepared through the hydrogel granules, suspension and distribution evenly in hydrogel content, and we mixed with glass rod, and finally we stored this in polythene paper. The nematode. Okay. And the second one is hydrogel. This is also, we can say, as the days increasing the storage or the survival of EPN is okay. This is the cost of formulations. We all also calculated. So we saw there 6 different formulations we prepared, and EPNs are the study evaluated that hydrogel, sponge and cocoa pit are the where we got maximum survival and infectivity. And in hydrogel formulation, the more than 80% survival is there up to 60 days, and we achieved 100% mortality at 72 hours to up to 30 days. The cumulative mortality remained high in this formulation, that is maximum 80% up to 60 days. In sponge formulation, it is peaked at 15 days 97% and declined 39 by 90 days. The highest larval mortality is 94% at 72 hours. In insect cadaver formulations, IDAs yielded at 15 days 100% mortality at, and cocoa pit formulation also there, that is more than 50% survival, and hydrogel and cadaver offers superior shelf life and virulence, and we can really through nice formation or good formulation we can use practicality in EPN in IPM practices. Thank you.
Thank you. A good presentation, very useful presentation. There is one or two quick suggestions. There is an EPN called Treponema species. They are more or less, they prefer thrips over many other species. So I would like, there are molecular sequences available for Treponema. I would like you to isolate whenever possible Treponema species, or try to get it from where somewhere it is available. The other one is, in the drip irrigation waters, now the habit is to put cadavers. So when you are doing a drip irrigation, when you, for this one you stop the micronutrients, put the cadaver and they go to the root zone. So in many other crops this is being, in banana and many others for pseudostem weevil and many others, we want to place very easily the juveniles very close to the root zone. So we are putting it in the cadaver, because in Orissa and Chhattisgarh you know, because
banana is not
pardon me?
Banana
Your entire VNR seeds is basically into drip irrigation. So you can talk to them and see how best VNR seeds can integrate with EPN, and also both based even in the nursery formulation as well as in the drip irrigation, there is a tremendous potential because the larvae move into the system.
Sir, हम लोग कोशिश करें fall armyworm is the major issue.
We are looking for a pellet formation along with BT and EPN, putting it in the whole of the plant, so that for the first 40 days you can manage. Already that systems are going on. Dr. Kaleshwar Swami and others are already to do this work, but it will work very well. EPN is a wonderful thing. I would also like you to request, if possible, to isolate the bacterium associated with it, so that if possible, the mortality that we can induce to see. After all, these nematodes, they cause only through septicemia. They inject the bacterium and then they suck up this thing and pass it on. So see if you can isolate the bacterium. And
sir, one of the scientists, actually Dr. Som Manishi, he is also a nematologist.
He is working on that.
Yes sir. उन्होंने बोला था सर कि अकेले bacteria cannot help.
Okay, let's look into it. Okay. Thank you very much. Nice presentation.
Thank you, sir.
Thank you, madam. We will call upon the other person who had to go. She has a. अंग्रेजी में कहावत है मैडम. हरी अप स्लोली का मतलब जानते हैं? Thank you, madam. Good work. Shield making, madam, quick. Silicone in. And defense, yes, Morer through, and all the entomology title, we would like you scientific name of the. If a pathologist and entomologist indicate the title, name, who else is going to put it?
Okay, thank you, sir. I'll take them. Good afternoon, one and all. This is Sana, Assistant Professor from Entomology Department of this very esteemed institution, presenting before you here. Yellowstone borer through major plant nutrients and rice. I'll try to stick to the time constraints as instructed by sir. Being a part of the agriculture community, I hope that we all are familiar with the and the menace. And we must realize that chemical pesticides is will create nation we are having. Will repel the beneficial element to the plant and then observation was mainly regarding to clarify this in terms of graph. Clearly we can see that diatomaceous earth at the rate 400 kilo per hectare and ash at the now we generally higher the medicine the effect. It is actually not true in this case. The higher doses of silicon, where as I mentioned, reactive oxygen species which are proline activation are also contributing anything on that. So we had two sets of observations. What promotes nitrogen uptake. Control value is lower, and in comparison to that, so these are the feature thrusts that I can say right now is that various doses of silicon, along with what are the nutrient interaction it can have, what are the molecular analysis we can have to justify our conclusions, and as well as the role of silicon transporter genes. Can we trigger them? These are sleeping genes that the rice plant is having. So any other transporter genes involved. So all these things are in the future thrust that I can think of. To conclude this particular presentation, what I am trying to suggest over here is we can go for application of organic sources of silicon like DA and RHA as we experimented on at the rate of 400 and 2000 kilo per hectare respectively. And this also helps in saving the environment and also conserving the natural enemies as well. And this is exactly what the silicon does, is not directly killing the species like pesticides, but indirectly making the plant strong, which will again repel the pest, helping us to save the environment. Thank you so much. With this, I would like to close my presentation. Thank you so much.
few good slides. Since you have such excellent skills, the entire presentation with hypothesis. Either put the hypothesis,
Yes.
you accept or reject later on. That will be a very good method of taking it forward.
Thank you so much.
There are one or two small things that need to be addressed. I would also look in the rice system. The garbages are becoming very slightly serious, and garbage is a plant response. So just maybe what to look into in addition to borers. We know silicon always wears out the mandibles in many of the lepidopterans.
Yes, sir.
But it's nice also to see how the garbages react to that. And as well as a small observations on BPH also will not hurt.
Sure, sir.
So these two things. Very good presentation. Thank you very much.
Thank you so much.
Thank
you everyone.
Thank you. Any questions? So if it is none, we will go to the...
Excuse me, sir.
Yeah.
My question is, how we can say that silica uptake can happen when it is controlling the stem? What are the symptoms in the plant?
As I said, first of all, first symptom is reduction in dead heart or whatever the damage symptom you are saying, and why exactly silicon you are asking, right? Why not other factors?
Silica uptake if it happens in the plant,
Yes.
what are the indications, any changes in the plant color, leaf color, stem stiffness?
Right. Whenever that's my personal experience, silica treated plants, you will easily get cuts on your fingers while going through the leaves, because the leaf blades become sharper, become sturdy, which shows that the silica deposits are making the cells stronger. The leaf blades are stronger, that is why it is very difficult to chew through them for the insects. Also the
stem borer mainly feeds on the stem. Then
Yes,
if you are
yes.
measuring the leaf, then how it will correlate with the stem?
Even the stem.
Long back in Erie, it wears out the mandibles.
No, sir. Actually, the stem borer mainly bores into the stem.
No, even then in the process, the mandibles get affected as the work of 30-40 years back itself. Erie has demonstrated.
The wearing of mandibles have been proved due to in silicon treated plants.
30-40 years back.
Yes.
Okay. Thank you.
Thank you so much.
Thank you. Next presenter, Meera. No, Meera sir. We call Dr. Rao, eminent entomologist, has worked a lot on pheromones and fundamental. Of course, time is 7 minutes, so we'll go. Yeah. Issue of semi-chemicals in modulating. Yes, please.
Good afternoon, one and all. Explore commercial crops. So I'm in Institute Rajahmundry. Here we have like turmeric, oilseed, and ashwagandha.
Come to the topic.
Yes. The become sucking insects, mainly the aphids and the whiteflies that also transmit plant viruses. Lepidopteran pests, it induce damage to the plant. The virus is very difficult to Sir, the insects are, so the communication is easier. Cases of in case of aphids, aphids is very difficult. Male type of or any type of pheromones, the communication is not it's not there. We have to make the plants to produce by the attracted by induced because it's only just a lecture. No, here some all compounds towards managing the identifying the attractive compounds for white flies. In the literature, green leaf and all these three compounds, xenol, state in practical applications. It's possible black ribs, retinal cultivation because it's not only the ribs, mites are also there. 2 to 1.5. Lakh rupees per hectare. There and some hesitate. They are also of Ganesh Tabashe. So we all know this. Okay, sir. 1 minute. We all know that how we can increase. There are 3 ways: through intercropping, raise a crop which attract natural enemies or repel the whole, that takes means not much practicing it, especially in only in that one ICP example was there. So that maize, and one more is producing the plants which produce natural enemies are producing the plant which which will not produce the compounds which are. This is one. When you see the technical readiness, application cost, and agronomic requirements, whose full strategy is operational scale, which full strategy using volatile organic compound, have to do need is invest. These were successful in greenhouses, protected cultivation, and such other where high expensive crops were there. But here, for the same type of system we can adopt here in India, we have to know. And again, when a crop is being invaded by a pest, and also we see natural enemies, when insecticides are not sprayed, that we see in the crop, but the same simulation can be done by us. The work we have done, because my colleagues were there, Kamal Jayanti was there, Shavaharan was there, all the people were there, all concentrating on pheromones.
Two quick suggestions that I would like to give is like this. Partition the sucking phloem, and because phloem feeders don't take and family wise different, and they are tremendous in terms of their own. There are trophic interactions, whether it is millibug or aphids or whiteflies. The role of honeybee in relation to the volatiles and tritrophic interactions. Second one that it is very much that the volatiles coming out of the honeydews itself is one of the triggers for the natural enemies to come or not, because they stay put on the leaf for a reasonably long time. The third dimension that we have to also think is, since you are a chemist, I am sharing that whether it is excreting a particular pesticide whether it is excreting a particular compound, can come very beautifully by analysis of the honeydew that increases the fitness of the bed. And for you have to separate it out from the thrips and others which don't excrete the honeydew. And the second one is, it will be nice to also to see whether the viruses are excreted by a process between vectors and non-vectors, and whether they are determined by these kinds. Since you are a good physiologist and a chemist, when we try to analyze the sugars in whiteflies, we sent it to Netherlands. Out of the 22 sugars, they were able to identify only 5 sugars. A large number of sugars even they could not identify, but they are playing a very critical role in their fitness, volatiles and tritrophic interactions. I feel those kinds of things, especially with Apis gossypii, with Myzus persicae, Apis craxivora, Memesia tabacea and such other species can tremendously contribute to test management. Okay? Thank you so much.
Thank you, sir.
Thank you. So we'll give a big hand. Nice work. You see, these are all basic research that is going to contribute to tomorrow's work. We call upon please, you are already there. So I am just calling persons because whoever expresses that I have some other work or I have to attend another session, I call such people. So please don't mistake it. So the please complete it in Gopal Swami.
Very good afternoon to you all. I am Gopal Swami from AP. Native and novel techniques for pulse storage. After hearing the president of AZRA, who is monitoring the bean in Australia, sitting in Canada or here also, this looks maybe primitive. And also after hearing the second presenter about many other smart techniques, and it is also interesting, they are also applying the sniffer dogs. So many people told regarding the food security. Mohandas Karamchand Gandhi, Norman Borlaug, and earlier it is only food security means to have adequate food, just filling the belly, but it's not so. It is having the nutritious food, and every country's goal is that is a second square yellow in color, that is zero hunger. And to deal with the nutritious nutrition security, legumes play a very important role, particularly in South India. Our day starts with legumes, and we are number one in production, also in consumption. To be self-sufficient, to be Atmanirbhar in pulses, the targets are very huge. So for this, horizontal expansion, vertical expansion has to be taken up. And one more thing is, majorly, as Dr. Jayas told, it is very much ignored, that is post-harvest management. So these are the factors involved in storage management and causing huge losses, starting from the moisture, temperature, insects, of course mites, fungi, rodents, birds, and other unscientific methods. These are all contributing for the losses. Coming to the pulses, the one and major problem is pulse beetle. Of course, secondary infestation of red floor beetle, lesser grain borer, rice weevil, rice moth will be there. Among the pulses, the 4 green gram, red gram, chickpea, they are more important, black gram. Among these 4, green drum is the most susceptible. And there are direct losses as well as indirect losses. If it is for seed purpose, even germination and viability will be lost, seedling vigor will be lost. And it is told, if you split the dal, you can minimize the this pulse brochette, but under pressure, even these split pulses can be attacked. This Calosophrocus can thrive on split pulses also. Yeah, since there are so many factors involving in the grain management, anyone say we have to go in integrated approach. If you say integrated approach, integrated grain management, normally if it is a crop, it comes host plant resistance, or here host grain resistance. But whatever the grain, say it is Bengal gram or peas and pea or whatever, hard seed, small seed, seed having thicker seed coat, and grain having phenolics or protease inhibitors, they are resistant. If it is so, if the grain is hard and seed coat is thick, it interferes with milling. If it is having more phenols, it interferes with taste. What we need is larger grain, at the same time better batter quality. So all these things may not happen if it is resistant. So these are some of the trials we have conducted, screened many varieties. Those are having high phenols are resistant. So here comes the novel technologies: IR radiation, microwave, ozone fumigation, hermetic storage, and biological resistant storage, cold plasma, smart packaging, many other things have come up. So we tried with splits, different types of dals, black gram, red gram, and chickpea, different packaging, triple layers, grain pro bags, aluminum pouches, ordinary gunny bags, cloth bags. We have tried several with all these three, and with grain pro bag and aluminum pouches, even after 90 days, we tried with infestation, artificial infestation and also natural infestation. They are well maintained. The microbial load is very less, moisture gain is less. Even the product quality, that is color retention, is also very good. If I say this is a smart technology, I would like to quote from the scriptures. Is there anything whereof it may be said, see, this is new? It had been already of old time, which was before us, says King Solomon. So we have documented some traditional techniques right in the border of Orissa, that is high altitude zone of Andhra. Even in Orissa you will find, in very Bhubaneswar, we have a museum, tribal museum. You can find some techniques there, that is hermetic storage, wherein we store in oxygen depleted condition. So our forefathers, they stored their grains in this hermetic storage systems. So this is another technique, same thing, in combination with ash. And they also keep these black cashew seeds, that is semicarpus uncut. They feel that it keeps away the evils as well as evils. So these coffee seeds, they are hung above the chula stove so that they are exposed to smoke. And the next one is that is the container of dried bottle gourd, and smearing the red earth, smearing the bamboo baskets with mud or clay. So these are all the traditional techniques. And keeping matchboxes, slime powder, these are all some of the traditional indigenous techniques. They may not know the scientific rationale behind it, but some of them works well. Even here, ashes and dung powders, they work well. Even camphor worked well, but it is short-lived because of it is an aromatic compound and it lives very shortly. So we have tested certain botanicals, sand layer, keeping 3 centimeters of sand above the surface, it will prevent. So we have extended, 1 minute sir, we have extended this technique to seed also, giving some little aeration as well as insulation. Extended to seed of peas and pea, green gram and black gram, and seed germination was very good. And coming to the oils, if you go beyond 5 ml, germination will be affected if it is for seed purpose, and also it brings rancidity to the final product. So these are some of the botanicals, that is Vasa. Vasa means aquarius. It can be exploited. Clove oil we have tested, and we made some botanical spice powder tablets also to prevent these bruises. And we tried with essential oils. Here, essential oil is important, its dose is important, and also the carrier material. Here we tried with plywood material. Here, not only the commodity production, consumer acceptance. Finally, that essential oil flavor should not be there in the dal or batter. So coming to the chemicals, many chemicals are, for many chemicals insects have developed resistance. This is one of the chemicals, spinosad is being tested. And we have many practical difficulties in phosphine fumigation in India. Still we are using these papers only and faulty sealing techniques. So coming to the ozone and CO2 fumigations, there are some of the studies. It is called controlled atmospheric packaging, modified atmospheric packaging. Here if you see, I had the one technique that above the grain space, that is in the head space, they lit a diya or they generate smoke before sealing this hermetic storage structure. So it is there already, CO2 fumigation. So infrared heating, microwave heating, there are several studies, radio frequency. So what I mean to say is, there are many techniques available, cold plasma treatment, but we have miles to go. Dr. Jas was presenting yesterday IoTs and about key sensors, CO2 sensors, temperature, RH, acoustic sensors, and smart control, decision support. They are yet to come. Again, these green storage principles and bio-based insulation materials, we have not yet exploited these microbial control methods, metarhizium, Bavaria, lecanicinium, and even the biocontrol agents and pheromone techniques, and even we have probes. So I think this should be the future of India. And finally, I will conclude that we have to go miles to achieve all these things, and you can contribute to their food security by not wasting your food. Thank you.
Thank you very much for a useful topic, considering that we import a lot of pulses and still our position is very precarious with 17 to 20 million tons oscillating, and depending on Canada and many other countries, Tanzania, quite a lot of import, and we are a protein deficient country and we require a lot of proteins in this. We have a lot of solar energy now.